I will start with introducing my team, who is hosted the Małopolska Center for Biotechnology. I do it now because I will then forget at the end. We have partnered with the Max Planck in Frankfurt, and we are growing and hiring. So if you're interested, you can approach me later on. But to give you a micro introduction to what we do, so we all know about proteins, and we have like 20,000 proteins in our bodies being produced. With splicing, we can ramp it up to, let's say, 100,000 different proteins. But this is not enough for all the type of tissues we have in our body. And how cells are dealing with that, they are further modifying those proteins with modifications. And these are the chemical adducts which are added to the proteins after they are made. It could be lipidation, ubiquitination for destroying the proteins, phosphorylation to activate them, and so on. And one of the biggest by mass of the modifications is glycosylation, which is addition of sugars to the proteins. And sugars can be even like half the mass of the protein itself, and they are extremely dynamic, and they are put on the proteins, and they modify the quality control of the proteins, cell recognition. They help folding of the proteins, interaction between proteins, and so on. I will not bore you with the details, but the pathway to synthesize them is extremely complex. They are made in Golgi and ER, and this is a kind of a stochastic process, which means that you don't get two proteins which look the same. The glycans that are put on the surface of the protein will be always slightly randomized and slightly different. And what it looks like. So if you ask somebody in the past what the protein, let's say a membrane protein here, for the sake of argument, is the SARS-CoV-2 spike protein, what the protein looks like, maybe 20 years ago, people would come up with this idea that you have this protein just sticking out in the membrane. And now with, let's say 10 years ago, people already know that there are modification of those proteins, there are glycans attached to the surface, here shown in green, but without looking at them, how they move, it's like trying to understand how the windshield wipers work without running them. So you don't know much, you just see some static structure. But if you switch on the dynamics and you sort of accumulate the positions of these glycans over time, you can appreciate that they cover the surface of the protein nearly completely. And the end product of that is that the spike protein is very efficiently hiding the surface of the protein from our immune system, only leaving a little bit here and here, and this is where all the vaccines and our antibodies are directed. Everything else is covered. So this is one of the tricks that the viral pathogens are using to hide themselves. And how we can see that? So we can basically use molecular dynamic simulations to see this dancing of the proteins, but they can also see the dancing of the glycans shown in green. So they are constantly moving around, and in one single frame, the protein is not completely covered, but if you accumulate those motions, they are covering the surface. And from this kind of simulations, you can infer like how the spike proteins are moving, how this is important for the infection itself. But you can also try to understand what pieces of the surface of the spike protein are accessible for antibodies, and you can predict the epitopes, which means you can generate maybe new vaccine constructs. And if we did that, we looked at the surface exposure from behind the glycans. We predicted some epitopes already in the beginning of the pandemic, 2021. And all of them by now were confirmed experimentally. And very interestingly, we could also see how the virus itself mutates. So all the majority of the mutations are actually happening in those places which are exposed, which means that our immune systems are exerting pressure on the viruses, and then they mutate the surface which is exposed, which is a target for the immune system. So this is not only a thing with the glycan, which is interesting, is also the drug discovery, because the glycans are covering not only pathogens but majority of the membrane proteins. Here you have an example of GABA, which is the major target of all the sedatives that we are using, or the painkillers. And the glycans are also localized very close to the binding sites for the drugs, and they are sort of overhanging over those binding sites, which means they will affect the binding itself. And so far they have been completely neglected from the drug design. So this is a new avenue which I think is kind of interesting. But why the structural biology so far was not able to really look at the glycans? It's because what I showed you, they are very dynamic, and if you average many positions of the glycans, as you see in the cryo-electron microscopy, for instance, if you take the average, you don't see the whole glycan because it gets completely blurred out. It's like you would take a long exposure photo on the main square in Krakow, and you average out all the motion, the main square will be completely empty. And this is exactly the same happening with the glycoproteins, that because they move so much and you average all these motions, you don't see them anymore. And this is what it looks on the molecular level. This is like a 1 microsecond animation of what the glycan is doing in time. And you can see this like spreading all over the place. And it's because of the chemistry of the glycans. So we can use simulations to access these motions, but they are very expensive. So the simulation I showed you, the dancing spikes, that was 20 million hours on the large supercomputer. And we were trying to figure out if we can make it a bit cheaper. And essentially we noticed that the proteins are moving relatively slower than the sugars themselves. So we designed, I call it the Born-Oppenheimer approximation for glycans. So it means that we neglect the motion of the protein, we just graph the sugars around them, and we created a library of simulations of sugars alone, and then we just put them back on the protein, and we see if we can predict what the protein looks like when glycosylated. And we could get a very similar morphologies of the glycans as in the full simulations, but instead of 20 million hours, we could do it in 10 hours. So it's significantly faster. And we can predict the same kind of epitopes, which means that if you have a new pathogen that attacks us, maybe we can just run the predictions of sugar and the surface exposure within a few hours and know which site should be targeted for vaccine design, which makes it an interesting avenue. Not only protein binding is affected, also protein dynamics is affected by the sugars. So if you imagine that some protein is moving around and there are some obstacles on the way, which could be the membrane, for instance, like you can see here, this is the beginning of the spike protein anchored to the membrane. It could be a multi-domain protein. If you put the glycans at the hinges, they will exert entropic forces, because when you bend, you reject more and more of the conformation of the glycans. So the entropy will try to straighten them up, and they act like entropic springs, which means that the multi-domain proteins they tend to be more straight, and the membrane proteins tend to sit more upright in the membranes, which is also an interesting thing you can predict. So this is the outcome of this problems with the solving of the glycoproteins I told you about, is that if you look at the protein data bank, if you look at the glycans in there, the majority of the glycans are composed of only 2 monosaccharides. So the peak is around 2 monosaccharides, and these glycans do not exist. So the glycans, real glycans, start around 10 units, which means that our database, which is used to train AlphaFold and all the structural prediction models, is flawed, because we don't know what the sugars are doing there. So it transpires into predictions, for instance, in the AlphaFold, where the proteins are predicted more compacted than it should be, because the AlphaFold doesn't know that the glycans should be pressing around and pushing the domains. Can we help the cryo-EM fields? Yes, we can to an extent. So if you look at the protein cryo-EM map in gray inside, and then you look at the unprocessed map, which you would get from the experiment, there is full of like hollow spaces. Now if we use glycoshields to model the sugars inside of those hollow spaces, we can fill them in completely, which means that we can maybe help the cryo-EM studies and understand how the glycans are moving, covering the protein surface. And we can use the cryo-EM data to guide our grafting, so we can take the glycoshield ensemble and we can filter it so that it fits the best to the electron density maps or to the SAX data, for instance. Since we are very fast, we can combine this with high throughput studies like with mass spec, and here is the case of all the human coronaviruses, which basically our collaborators, we could model and look at the surface coverage of them within one afternoon, let's say, instead of many months. And what I told you is that if you look at AlphaFold structures, in many cases, I mean, they will predict it naked without glycans, and in many cases they get too compact. They are hinges which normally would host glycans, and yet they are very close, like a folded. And we know that if you put the glycans back in, those close contacts should not be possible. So maybe using the glycan information, we can remodel those proteins. Cool. So what we are trying to do, we are now trying to combine the AlphaFold predictions with glycan modeling. And these 7 human coronaviruses, it used to take us basically 3 months of preparing the model and then months of simulations on the supercomputer. And now I could do all the 7 models of the spikes of the coronaviruses in one afternoon. So we actually achieved quite a speed up here. And so we have, I hope I convinced you, we have a high throughput opportunities for high throughput modeling. We can try to design vaccines or drugs, including the information about the sugars. We can maybe help the experimental pipelines, maybe retrain the models like AlphaFold, which then will allow us to maybe make personalized proteins, because the proteins in different humans will look a bit differently because the glycans will be different. Also in different tissues. In the cancer cells, the glycans will look different. So the proteins that you take, the vanilla proteins from AlphaFold will look differently in different situations. There is a research and commercial potential, but there is also challenges, especially in Poland and EU. We still need HPC resources to run our predictions, and if we want to combine them with tools like AlphaFold and other AI tools, we need some platforms to put them together, not to hack it with the bash and then try to copy them around. The same with the solving the cryo-EM structures, how to connect all these things together, how to gather the data, because maybe we are dealing with the patient data about glycosyl transferases, and to know what kind of sugars are put on which people. There are very specialized diseases which are very rare, and knowing those glycan composition can lead you to the patient directly. So there is also a question how to store the data in a secure way. And also, if you want to commercialize, the scene in the EU and Poland in particular is quite fragmented and unaware of the solutions that science is providing. And the expert base available in the AI is still quite low. In Poland, it's estimated that we are missing 25,000 AI experts at the moment. This seems to be not only the problem in the structural biology, but similar pain points have been observed in healthcare, language models, and the space sector, about which we heard about. And this was actually studied by the Ministry of Science recently. So here is the answer that the EU is trying to provide. There was an AI Act which is trying to introduce AI factories, which would provide multiple layers of integration and to providing platforms for studying HPC and AI tasks for both commercial and research entities. And I have to say I stole those slides from Marek Magryś from Cyfronet, so don't blame me for that. So the goals of the Gaia AI as a project is to first of all develop the infrastructure, so to build supercomputers where we can actually host all these things we want to do, to increase the competitiveness of our markets within EU but also worldwide, so that the EU is not depending on the solutions from other continents, which might turn out to be hostile at some point, which was not the case in the past. So we also need training to maintain that, and we need what EU calls a trustworthy AI. I didn't know, I had to look it up. So this is the AI which produces the consistent behavior. It's interpretable, is not biased, it protects the data, and is significantly documented. So this is the idea that they are pushing for. And the money which was granted from the EU is to build such a computer which would be integrated into the Polish AI and HPC scene. So Gaia comes as a middle layer. So at the bottom you have the typical resources that we are using on the daily basis, Cifronet, ICM and so on, with the supercomputers there. And they are at the moment in existence or in planning 2 AI factories on the national scale, which is Piast and Gaia, which are trying to sort of not compete with each other, but rather collaborate. And then for the even bigger deployments, which require even heavier resources, the idea is that we will be using Lumi in Finland, which we are stakeholders as a country as well. So the Gaia architecture, it's focusing on the called hard sciences. So its main focus is health space, and a big chunk is large language models. And the idea is that the Gaia AI would provide services for researchers, for students, but also for the small and medium enterprises and startups. So this is a hybrid solution that I think is very interesting and might help all of you that want to develop your own companies and develop new AI models. It should provide tools and services which should allow you to train and deploy the AI model, the whole pipeline, using the guidance from the experts. It's also heavily depending on the user support from the experts that would be hired in Gaia, which will organize webinars, hackathons, and different meetings to facilitate the training. And there is a number of different access modes. I guess it's gonna. I mean, we don't really know everything yet because it's still in the making. But there will be access for the researchers. There will be different grants for the startups, and then all the way to the deployment on the free market. And the data management is also interesting. So it goes all the way to the data spaces, and data spaces is also a very fun new feature which EU is trying to introduce. Is that the data is distributed across the countries, is not centralized, but is following the same structure. So you can access the data from wherever you are, from different institutions, and it's usable in the same way everywhere, which is, I think, it would be very useful for training AI models. So the consortium is formed by the big supercomputer centers. There is also, like the main chunk of that is Cifronet. There are 3 centers, which is a mother and child center, Sano, with people representing it somewhere here in the audience, and Małopolska Center for Biotechnology. And these are trying to produce the core services, which will be starting from protein function prediction to protein structure prediction, all the way to digital twins of disease spreading and training for even training for surgeons and things like that. The deployment plan, I think, is interesting because we are at the beginning of the path. The grant was granted, but and the whole budget is 70 million euros, and the funding will be split 50-50 between Polish and EU sources. I think the most important part is that this is a long-term maintenance. The operational costs are 80 million euros are established for 60 months within Cifronet, so it's going to be a long support. So we hopefully will be able to use that all the way to 2032, which is the typical lifetime of the machine. But I would not expect this to be operational too soon, because we need to get all the procurements done, and let's say annealing procedure of the cluster. I think it's going to be 2027 where we actually start to have some production runs on the machine. It will be hosted in Cifronet proper. There are rooms ready waiting for it, so I think we will see some movement there. And what we are trying to do here, I mean, we are already working on the new deployment of GlycoShield, which will be available within the Gaia AI. which will be coupled with the AI models like AlphaFold. We have extensive data sets of glycan dynamics, which we can use to train AI models. So maybe we don't need to even do the grafting anymore. Maybe we can just predict the properties directly from the composition of the glycan. We want to integrate the cryo-EM pipelines. So maybe it will look like that. And I should end with saying that there will be open days of both CitroNet and Gaia happening on next Monday and Tuesday, and you're encouraged to sign up.